Pathogen testing is what we do primarily. I do pathogen testing and do sales for companies all over the US. We handle everybody from small craft growers, home growers, to the largest MSOs you can think of. And we help them grow cleaner. We help them grow more efficient with pathogen testing. So keep us in mind. That's what we're here to do primarily. Secondarily, and kind of the subject of tonight's talk here, is our genetic fingerprinting services. Genetic fingerprinting is a pretty new concept in cannabis. It's going to solve, it has the potential, hey, look, it's me. Woo, all right. It has the potential to solve a lot of, let's just say, complications that exist in the supply chain currently. And, you know, it's just like, it's a really great tool that we developed. Basically, Dr. Angel Fernandez came up with a panel of 3,000 markers on the cannabis genome that are meant to be trait agnostic. It's simply only for validation and identification. So we're avoiding the issues that people might think people might do with this type of information. Just like your fingerprint can't tell you what color your hair is, but it can identify you individually. That's what our fingerprint does too. So if you're a cannabis breeder, you know, the minute you think you're going to give a clone to somebody, it's probably a good idea to find a way to objectively measure it and be able to go back and validate it. That's what our fingerprinting can do. If you are a tissue culture nursery and you want to help yourself unfuck mix-ups that might happen in vitro, if you have an objective measurement of those cannabis genetics online, you can validate against those to help clear up mix-up quickly. If you're a cultivator and you want to make sure you're getting the same cut from a nursery 2 years after you got it from them and they're telling you it's the same one, if there is a transparent chain of custody with the fingerprinting, there's another application there. I mean, it keeps on going on. If you're a consumer with a medical condition and you want to make sure that you're getting the same genetic that helps you in the way that you know nothing else has, and you want to make sure nobody's just relabeling something, this is a great way to do that too. That's pretty much it. I just wanted to give that out there, and you can see there's a lab, yeah. Breeder, tissue culture lab, nursery, cultivator, all the way to consumer. Validation is super important here. This is going to help tighten things up across. It's going to also give breeders a way to give some teeth to contracts that might have licensing their genetics to growers. Locks keep honest people honest. Same idea here. That's pretty much it, because I'm not a science guy, and luckily I work with a handful of PhDs who are incredible science people, and some of them are going to come up right now.
So this, hold on, before we jump into that, I want to go through the phylogenetic matrix, because
this
was
oh my God, yeah,
the LCG Battle Royale that we did last month in Los Angeles was awesome. We saw that there was 3 different cultivars that were labeled LCG in the competition, okay? So, Matt, what do we learn from this?
So this is pretty fascinating. So I came from background working in nursery operations for a couple of years. I've worked with a couple of other cannabis genetics companies over the years. And this kind of exposes some of the questions I've always had. So this is fantastic. Like Sergio said, all these cuts were fire. This was all amazing weed that everybody put in there. We took samples of all those 10 submissions to the LCG battle. We did genetic fingerprinting on all of them. And out of those 10 submissions, there are three genotypes within there. So we had 6 cultivators that all shared one genotype, 2 sharing another, and 2 sharing a third. But they're all LCG. Awesome. And like Sergio said, this is not a matter of like, what's the real and what's not the real. This is a matter of, hey, genetics affect everything about a plant's performance. Genetics affect everything about consumption. If you find a cut that's really good, the most actionable data for you is probably not going to be what somebody called it. It's probably going to be what the genetic fingerprint is. That's it, man.
And what leads to that final end number, that ticket, right?
Yeah, it gets that ticket.
Am I going to invest 4 months into growing something that I don't know if it's the real thing, right? Is that the same thing that I'm going to go shop for? Well, we have a tool for that now.
It's pretty cool. It's cutting edge stuff. I feel like I work in a sci-fi novel most days, so thank you all so much for coming out. It's been a pleasure working with Sergio on this, and with Lindy, obviously, our host here, so take it away, homie.
Appreciate you, Matt. Thank you. All right, so we're going to get up to our next stage panelists, and I'm just incredible to be sharing the space with such incredible people that have contributed so much to the industry. And again, guys, we're here to learn. We're here to level up. We're here to cultivate curiosity, right? We have a lot of questions about genomics. There's a lot of use cases that we can utilize this conversation to go, and I encourage that. So again, shout out to MyFloraDNA. First to the stage, I want to welcome CEO and co-founder, Dr. Angel Fernandez. Thank you, sir. Welcome. Quick name and intro, bud.
Good night, everyone. Thank you very much, Sergio, for the nice introduction. And we are a scientific company. We are, I would say, probably 50% of all our employees, they are PhDs with experience in many other crops, in genomics and molecular biology and genetics. And we all share the same passion, molecular biology and bioinformatics. So what we bring to the cannabis industry is our experience, our knowledge, and we are always welcoming folks from the industry that they have experience in classical breeding, in testing, and in other aspects, and combining the scientific data with all the other experience and cultivation and so on. So thank you very much, Sergio, for having us tonight, and we are super happy to answer any question that you might have related to genetics, genomics, pathogen testing, fingerprinting, and so on.
Thank you, Angel. Round of applause for Angel and MyFloraDNA, please. Want to take a seat? Next up to the stage, we've got the five-star general himself, Mr. Kevin Jodrey. Welcome, Kevin.
Thank you for having me here. Gosh, it's such an esteemed audience and group. It's kind of hard to say what you do. I just try to solve problems and issues in the supply chains, and I usually do it through genetic understanding.
Straight up. That's it. Appreciate you, Kev. Thank you, man. Last but not least, we got Mr. Todd Denkin. Welcome, Todd.
Thank you, buddy. Thank you very much. Hi, everybody. I'm Todd Denkin, and I have been in the analytical testing business for the last 9 years right here in Nevada. Prior to that, I was in the cultivation business and then the hydroponic business before that. I've been around legal cannabis since about 2014. Long time. Oh, no, 2009, so for 14 years. And a big fan of genetic identification for really ensuring it is what it is. Because we see a lot of it isn't what it isn't at the lab.
Thank you, Todd. Welcome. And from the tissue culture and genetics side, Justin Gibbons, Starts.
Hey, how's it going, everybody? I've been in cannabis my whole life doing traditional distribution. As the legal space started opening up and genetics became important, I started hoarding genetics and finding that they're very dirty. And looking around for people that had a solution, they didn't exist at the time. So I was forced to start my own tissue culture lab, and I essentially started hoarding all the genetics, cleaning all the genetics, and now I have a huge lab. I sell genetics to everybody, and I provide clones to as many people as I can to help all the growers out there that are suffering through the hop latent druid and other things that present risks to the grows. So,
welcome, guys. So innovation in cannabis genetics, a lot of cool things that we can talk about here. And I would love to just open up the conversation because you guys have a lot of great thoughts. We had a little bit of a pre-discussion the other day about this and how fingerprinting and use cases and all these different things kind of come about. We have a couple of topics up here. I just threw DNA fingerprinting, commercial viability, global gene banks, public health and safety, just as some key things that if any of these want to start off the conversation going for it. Let's go for it.
So my definition of DNA fingerprinting might be related to social security number or your license plate. Okay, so I think I guess 9% they will that every single plant, every single strain or every single cultivar has its own social security number. So that is the foundation of genetics. But we all should grow together in order to get the social security number for every single strain cultivar. Okay, so we develop a methodology based on DNA molecular markers that can tell you the social security number of every plant. So if you are growing Gelato 41 here in Las Vegas, if you grow the same Gelato 41 in Sacramento or in Illinois, it should have the same social security number. When it doesn't have the same social security number, there is a problem, okay? So we have those tools to let the growers, the breeders, the cultivators, the consumers, that they are not growing Gelato 41.
This comes back to a very key topic in genetics is trust. Okay, so trust and transparency.
It's trust why we have scientific tools.
Exactly.
We're all special.
That's a great point, Angel. So registration, crop registration, right? And Kevin, I think you touched on something that was really cool the other day about the bank and how do we utilize this kind of technology to expand into other markets, keep the culture alive with certain landrace genetics. Obviously, there's a lot of terroir and things that come into effect with that, but talk to us a little bit about how this can kind of apply to that and how we can work together on it.
Because cannabis is a global crop, what you have is you have a global community of cultivators that really have never worked together. So we've taken stock from all these other regions around the world, and North America is the last in the supply chain of genetic distribution. So we really have nothing native, and we have the shortest amount of time holding it. But we have the most market. And so what you really need to do is you need to understand that cannabis, by virtue of the fact that it was this societal movement and used in its entirety, that really connects people in a different way. And so the ability to work with all these other genetic resources allows them to bring in germplasm into our world that lets us take profiles and traits that are just proven and extremely hardy, and then allow us to be able to bring that awareness to the public so that a derivative, some form of payment can go back to the source. What you have is you have an ability to operate in a holistic way that's actually really successful for the operators because you're never stuck with a limited gene. And the reality is I work in so many countries that I realize that all craft cultivators, operators, they're all the same no matter where we go. So they're small in the room, but globally they're not small.
That's a really powerful statement. We're small in the room, globally we're not. And so this is kind of where this registration, this entire process of everybody communicating and sharing that's one of the key things, right? The social security number. So having the ability to share what we actually have at that point in time, like that's really impactful. So I know Todd's working on a really cool project called the Smuggleverse. And Todd, please elaborate a little bit more on this because this does go back to the genetic profile and what you're actually having in there in the Web3 space.
Sure. I mean, eventually, everything will have a fingerprint. I mean, it will be the way everything will go. And eventually, everything will be on a blockchain to verify ownership, to verify everything that you're producing would be real. So it is the way of the future, and you have to embrace it, right, or lose. What we're doing at the Smuggleverse is, we do custom digital assets of cannabis integrated into a Web3 game, which has a licensing aspect in the real world. So it's a complete Web3 play, but everything works on this blockchain to guarantee, play to earn from a Web3 standpoint, using cannabis as a commodity, as well as psychedelics, alcohol, and edibles, later on the plan. So from a DNA standpoint and from a marketing standpoint, it's the ability to create custom strains, which then you get the genetic thumbprint on, put it on the blockchain, and it lives forever. And you can prove ownership. The whole thing is protect my IP, make my deal with you, right? Not my deal with everybody, right? So it's the only way to protect it and to really prove that it is what it is.
And protection is a very, very tricky thing. And Justin, I want to tap into you on this one as a genetics global distributor. You work with a lot of breeders. And how does that trust and that protection and what you do in the tissue culture lab and how you go through the distribution side of that? Talk a little bit about that with us, please.
Yeah, sure. So a lot of the breeders I work with do a lot of hard work, right? And they sweat, blood, tears into genetics. And, making deals with all these new companies, right? Looking at the legal space, enforcement's a real problem, and tracking that and proving that something is what somebody says. But if a breeder takes something in to a big company and wants to do a licensing deal, right? Sometimes I've seen things happen where there's a fallout, right? They keep the genetic, they rename it, and then they keep pushing it forward. Now that breeder is getting fucked, you know, and not making that money and did all that work and that hard effort. And in traditional ag, this isn't a problem. Royalties are a real thing, and fingerprinting, everything, it's all normal, you know. And cannabis being, you know, illegal for so long and taboo, right? A lot of folks didn't want to work with it. A lot of scientists still, you know, are afraid to move into this space, right? And we just need to create a space where everybody can operate legally and be taken care of. And trust, but verify, right? And verify that people are paying you right. Verify that the genetics are what they say, you know? That's a huge thing. And with the genetics that we have, right, that I move around the world, a lot of people have different ideas of what a genetic is. Sour diesel, for example. I can't tell you how many people ask me, oh, the sour diesel's coming back, like I want sour diesel. I have 2 different sour diesel cuts, right? Which one do you want? Do you want the actual New York sour diesel, or do you want the one that got popularized on the West Coast and then moved to New York later on, right? The big Galaxy, you know, long leaker, or the one that's short and steady, more OG, right? And people, they really, they don't know. And so I have to talk them through it and talk about that, you know. It's interesting.
I think you look at gelato as an example, right? So over the last 10 years, gelato is the most impactful variety in the industry. If the U.S. industry is worth $20 billion, gelato is probably in 20% of the genetics that are sold in that. That means it's a $4 billion a year variety. Did Mario get paid for that shit? Oh no. So that's the issue. That you develop a product, no one really knows the reach it's going to have. But if you have a fingerprint that is marked it, and he's able to keep it in some form of protection that says, I own the genes, anytime anybody used it in a hybrid, he would be able to genotype it and show that that was his source material used. And that's the problem, is that you're taking people who come up with some incredible shit, and then you don't pay them their proportionate worth. So what really is that plant worth when it's probably $4 billion a year? So over the last 7 years, we'd say $30 billion have been sold to gelato. So like that's the point. And when you talk about numbers like that, a couple points on it and ownership is massive.
And that's a big part of it, guys. Why haven't we adopted some of these tools from traditional agriculture, right? It's scary. It's scary to give up your genetics. This is where the whole trade agnostic discussion about the Battle Royale came into play that Angel has this chip for. When we talked about this initially, I'm like, dude, I can't have a chip that's going to have as much data in there that can potentially lead to a concern of somebody stole my genetics. Trade agnostic. So that was one of the things, Angel, thank you for listening, because this took a couple years to really get to that point of having enough trust and confidence to be able to do something like this. So I appreciate you. I commend you for having this tool, because this is a great point Kevin brought up. There should be breeder royalties. And this is a big thing that we have to start talking about. Intake protocols, Justin. We were talking about what does it mean to bring in a genetic, do a CNR on it, bank it, store it, cool. But what about you fingerprinting it on the way in? That way when you fingerprint it on the way out, you know it's the same thing. How many times have you gotten something that they thought was the genetic that they were getting CNR'd, but actually wasn't?
Oh, yeah, multiple times. I've had people call me like, hey, you gave me back the wrong thing. I'm like, well, you gave me the wrong thing then. Your employees screwed up, right? And recently we did that R&D project, and we were able to truly prove, like, one very well-known breeder that gave us something, or his employee gave us something, right? And I was like, you know what? No, like everything coming in the lab needs to be fingerprinted, chain of custody controlled, right? So we sample it before it comes in. That way when it goes out, we confirm, hey, you're getting back exactly what you gave us. So out of that, you know, it's a liability thing on our part, right? We don't want to get blamed for doing something we didn't do. And that became a big thing. And so it's a powerful tool just for us in that aspect. We save a lot of money on liability issues because of that. So, yeah.
Liability. That's a big thing with genetics, right? So not only from a trust perspective, but if you sell the wrong cut or somebody claims to have the certain cut that you're about to go and invest months into, It's a pretty big missed
This
investment.
is something that I would like to bring up. We have been talking with many tissue culture labs, many nurseries, and we were told that 5%, it's a big number, 5% of the cuts that they send out, they are mislabeled. 5% mislabeled, okay, by human errors. That happens, it's normal. So right now we have the technology to detect these 5% of plants that they shouldn't be in the same lot as they are sent, they are shipped.
And are you just taking a little leaf sample in a vial, and how long does that take to process?
For us, it takes us the process, we do everything in-house, okay? So that is one of our main things, that we do every single analysis in-house. We don't outsource anything like other companies. But the process, building the DNA library and doing the sequencing and so on, it takes 5 days. So within 1 week, the tissue culture lab can know that 100% of the plants belong to this cat or to the other cat, and they are not any mislabeled plant.
And recently on Hill, you showed me something that you can homogenize all the different cuts in that
Yes,
one vial to see
yes.
if you have a miss, right?
We are always, so mission is trying to leaders, growers, and so on, okay? Trying to make clients' life very easy, and obviously to be economical. So with this statistical analysis, the tissue culture labs, they can pull, I remember, I think, 15 samples, 15 clones into the same collection tube. And with that information, we can reach out a 98% confident interval. So it's very high in statistics.
That's powerful, because now you don't have to go individually, but you can homogenize it, save a little bit of money on there, because this is a costly process as you start to go through it at scale, right? And so, Justin, you touched on your intake and exporting, or the in and out process of a TC lab, right? There's a lot of liability in that, quick turnaround time, because we're all looking at the time it takes to use testing or to use any validation services to begin with. And that was one of the reasons why I loved working with MyFlora. I was working with 3 labs, maybe 4 at any point in time, to do my regular virus and virate testing. Right, as a nursery, you got to have a consistent pathogen screening schedule. So the plant is getting screened every 3 to 4 weeks. So the cycle, the time of it, I know that if I have my results back within a day or two, cool. I can make sure that if there is a hot batch, I can retract that. I can make it better. I can tell you as a cultivator, Kev, make sure you don't move these stakes around in that tray. Let's test the plants. I tested them on this week. You test them on intake, so you can homogenize those. If there's a problem, let's talk about it. Let's get that replacement going. So there's this trust and accountability factor on both sides. If I know you're taking full responsibility, it's a latent viroid, right? We don't know if it's there or not. Again, it could potentially come out within that 3 to 4 week cycle of our testing cycle. It could. There's a potential there. So again, it's one of those things where time is one of the factors that we can use to build more trust and build accountability along the supply chain. Kevin, as a longtime propagator, I know you've seen a lot of different iterations of professional maturity in cultivation partners that you work with throughout the years. And I think that's one of the biggest ones that, from my perspective as a nursery operator, it's about the relationship management aspect of who you're selling genetics to.
It's also, you're going to make a mistake, right? Because it's a living entity. So people, animals, plants, there is no control that's absolute. It's not a widget. And so because of the error is going to have some statistical level of flaw. And it's trying to minimize that and being able to slow the spread of it. And that's where having, good lab analytics and people that can help you refine it and get the price down so that you can actually afford to use the tool. Then you're able to say, okay, we made a mistake, we can tell them and we can fix the mistake. But there's no perfection in living things. So this relationship has to build together to make it to minimize the issue.
Correct. Understanding that cost factor, right? Budgeting for testing. Budgeting for all these things that you managing a P&L, you should be responsible for. Understand your forecast, guys. Thank you, guys. That was something I've always wanted to like talk to you about, Kev, because I know that we used to buy genetics from you when you were at Wonderland, and we had our 215 nursery in Sacramento. Like, I was always impressed with a lot of the things and the serviceability. Even back then, you guys held at a very high standard. So thank you, brother. Appreciate that. Set the stage. Guys, I just want to say, if you can please keep it down in the back. It's super loud up here in front. So if you want to take your conversation maybe to one of the other 4 buildings here on the property, that'd be super dope. Appreciate you guys. So guys, Q&A. I want to jump right into Q&A because I know we've got a lot of interesting topics over here. I'm going to start us off with Mr. Chad from Maven.
So, I'm not a geneticist at all, but how similar is cannabis plants? I mean, what's the biggest difference in genetics you see between a gelato or an OG? Like, what percentage of the genome is different? I want to know the most different plant. And 2, I want something for Kevin. Of plants, have you found the origin of a cannabis plant? And has it, per where you go, do you see a trend on expressions of cannabis?
So the first question is, depending the wild species you add into the phylogenetic analysis or land races, okay? If you say me 3 gelato 41, obviously they're gonna be 98, 99, 100% identical. If you add a Mexican Osaka land race, then the calculations, the algorithms, they are gonna be completely different. So we can get 50%, 35%, 40%, depending the variability that you are adding to the genetic pool.
Like Pakistan is populations that were in like the 1500 year zone, where the herb is in these regions for millennia and a half. But what you have is you have feral populations that really let you understand what feral cannabis look like and how we steer it, how we selective breeding and handling, we were able to refine and develop it. But what you find is, when you're looking at cannabis globally, what you're understanding is metabolite packages that are formed in stress responses to specific environments. And so it allows you to start to really understand this bigger picture of profile, and then how do we drive the cannabis into that direction equivalent. So like you have the soil analysis, it's a sap analysis, it's a DNA analysis, and it's cultivation steering. So you can try to get how do you plant perform in that area that makes it so desirable, and how do you make it give that similar performance in your hands. And it's fascinating because at the end of the day, it's hard. So like it's always humbling, always trying. But that's where I think you need global work. You need people from all over the earth, so that what it does, it allows you to better understand attributes and traits of desire and interest that you want to put on your process in your plants. And how do you do that to get an effective result? Because if it doesn't sell, or it costs too much to grow, or its resistance is low, it's death as a farmer. So all these agronomic considerations, and then consumption considerations, does the consumer really want it? And then can you actually grow it? And once you get that trifecta in play, you're in.
Great question, Chad. Thank you. What other questions? What questions do you guys have for yourselves?
Oh, that's a good question, man. You got a panel and nobody's asking any questions that are skilled.
We got another question. Okay, we got one right over here.
Hey, Devin, Michigan again. So what kind of utility is there aside from like protection in DNA fingerprinting? Can we like see, resistance genes? Can we see like susceptibility? What kind of things can we use? What utility is that?
Okay, so other crops tomato, if you are a tomato breeder, if you are a almond breeder, if you are a cherry breeder, if you want to protect your invention, you need to provide a list of morphological traits. Okay? Those morphological traits, they are gonna be the foundation of your invention. Okay, phenotypes, exactly. On top of that, now the plant variety agencies, they are adding an extra layer. And that extra layer is based on DNA molecular markers. At this point, there is not a regulation that you need to show, okay, my plant is, you have to, so you have to demonstrate that your plant is powdery mildew resistant based on phenotypic data, based on inoculations that you have done in your greenhouse. But you don't need to do that based on molecular markers. However, you can demonstrate, you can add an extra layer of protection based on DNA molecular markers. That is the social security number of your plants. But they don't need to be associated to any particular trait of interest. Is clear? Yeah.
But like, if you're able, say like, so like me, I'm not a scientist, but I work with science teams. I help science teams understand what it is we want to achieve. And so the ability for me to say, this is what I would like to see, this is a problem. We're going to talk about ratio of A to B buds. And I'm going to say that an average plant gives me 32% A quality flower. And I don't want to have to do a tremendous amount of work to achieve more. I need to do it genetically. So if we can use marker technology, then it allows us to start to understand that the plants that exhibit the traits, can we identify the marker that controls it, and now can we steer towards that in the breeding cycle so we can intensify the traits. And for us, it allowed us to go from 32% A flower to 50% A flower just through breeding. And so it's that kind of stuff that when you start to dive into it, what it does is it's a process, so it takes years, right? It's not fast. It's not like, oh my God, I understand it right now, I'm going to do it. You have to locate it and actually find out what happens when it's used and what other genes are triggered by that gene. And so it's this process, but the point is that if you're able to utilize scientific work and you're able to look at it from a genomic perspective, you're able to start really steering and driving the results you need as a farmer. So if we just had better seed stock that gave us a 51% A flower versus a 32% A flower, we're doing better. And like, for me, I'm a farmer primarily, right? So like all the gene work I do is other stuff, but like, primarily I'm a dope grower. So that's what I care about. And that's why it makes it easy to work with science teams, because I understand what I want to have happen. And I know what happens if the traits of interest are applied. So that's where your marking comes in, because eventually you should have your own variety that you work with that allows you differentiation in a very crowded market.
Real quick, I'm going to jump back in. I just want to bridge back to the original question a little bit to help. So fingerprinting is a way of identifying specific areas in the genome and comparing to each plant, right? So you can see what's similar and what's different. It doesn't have to do with traits in the way you were talking. For trait identification, you have to do full genome sequencing, which is much larger, much more expensive, a bigger process, right? And so to do that and leading into molecular assisted breeding is kind of what you're talking about, right? It's a whole different area and a whole different process. Fingerprinting is really good for identification and enforcement of genetic itself, but not necessarily trait identification specifically. Those are isolations outside and, again, going to full genome sequencing, which is a much bigger, more expensive process. $2,000, for example, around to do a full genome sequence versus a fingerprint is like $100, $200, right? So very different.
You are totally right, Kevin. Depending the main goal of the breeder, or the cultivator, or the nursery, what we had on mind when we developed these technologies to help as many players in the industry as we can. If you want to do marker-assisted selection breeding or molecular breeding, and you want to target the specific genes, we can help with that. We can design those markers for you. But the overall industry wants this kind of technology that is enough to differentiate one cultivar or one strain versus the other. So for that, we don't need specific genes related to specific traits. We have other technologies. It can be whole genome sequencing, it can be genotyping by sequencing, or even can be gene editing and CRISPR. Okay, but the goal of this technology is to help growers, breeders, tissue culture labs, and also consumers. Because if you are, so if there is any specific strain that helps your health, and then you change the dispensary or you change the state, and you want to get the same cultivar or the same strain because it really helps your health, unfortunately today it's not happening that. And it's not happening because there is a change of the name somewhere in the process. So with the fingerprinting, we can authentify, we can protect, and we can tell to the retail, to the consumer, oh, this is Gelato 41. This is the real Gelato 41. So if you want to solve your anxiety problems or whatever, that is gonna help.
So kind of to build off of that, have any of you identified a dominant or recessive gene type like what we see in humans? Like, is there things that we can see from certain strains that we can maybe predict that it's going to come through on our genetic if we cross it with it?
When you're running a breeding project, if I can find the markers, then what it does, it lets me look through a large population, and it lets me say the 10,000 plants I looked through, 63 of them had that gene present. That lets me know that those are the only ones I can use in the project. So it just simplifies it. And like we did this for a long time. You're trying to find the frequency. What is the ratio of numbers needed to get the progeny desired? But with sequencing, it makes it a lot easier. It's just that it's expensive. But the thing is, when you have good genes, you have something of value. You got to be able to figure out how to leverage it and work with companies that have the ability so that you're able to dive into it and prove it. Because things that have high palatability, human attraction, they really don't go out of vogue. They move in cycles. But if you can map and develop, it's sellable always on a diminishing market and you scale down your overall canopy. When people like something about it, gelato's a good example. I use it because it's so hot. But what did it do? It split like a stop. And then it became hot again and again and again and again. So you have something that's worth $30, $40 billion over its lifetime. If you have stuff that has those type of profiles and those qualities, you want to be able to identify what made it do that. And you can't do it as an individual. I have to team up with somebody like Danielle. And then it's held in the TC form. So it's about like what are you trying to do and how do you use the scientific tools to the level you can afford. And if you can't afford it, how do you form a relationship so you can?
That is.
Is this on? My name's Anthony from Modesto. A couple years ago, they were talking about taking the best of these traits from the visual to the data and combining them into a synthetic seed. Is that a practice that anybody's practicing right now?
Synthetic seeds specifically are just a way of preserving already existing material and either cryopreservation, through refrigeration, or field implementation. It's not like, there's a lot of misinformation constantly in cannabis. It's unfortunate, right? But synthetic seeds are a real simple thing. So I wish I could show you a picture right now so you could visualize it, right? It's not in the way you're thinking.
It's like a meristem within a little capsule or something like that.
Or really a node, right? Or some established plant material specifically, right? It's a real simple thing, but it's a very hot topic right now online. People are like, oh, you do synthetic seeds? And I have to explain, well, they don't really have a lot of purpose to me, right? Everybody in this community wants their plants fast. Synthetic seeds aren't, they want local. Yeah, real fragile, yeah. The thing that they're really good for, though, is smuggling genetics into a country that otherwise doesn't allow that, right? That's the best thing. So, everybody wants a specific clone, and with seeds, you can't have that, right? So sneaking in bigger clones through airport security, things like that can be a problem. But if you have a little vial synthetic seeds, put them in a little pill bottle, right? You can get away. And in countries that it's completely illegal, you can then smuggle those in, have that clonal variety that you want. So that's what they're really powerful for.
So if I could touch on that, as a tissue culture scientist, okay, there's nothing more important than separating that meristem occasionally from the plant. Because once you separate that meristem, you've basically taken away all of the history of that plant, and you've allowed yourself to start back at the beginning of its creation, when you first took the cookies or the chem dog or the Cuban black and you crossed it into that other plant. So by getting to that meristem, you allow yourself to reset that genetic to its truest form. It'll express itself in ways that you couldn't even possibly imagine, because you've removed all of the pathogen resistance, which stands in the way of all of that production that you're looking for. The panels that we're talking earlier, we're talking about SOPs for crop steering and other types of advanced techniques for growing this hypersensitive light plant. But what's left out is all of the pathogens that are standing in the way of that development. Tissue culture is the first and foremost way to remove those pathogens so that that plant can express itself without, it's almost like being a land race in a cross.
You know what's weird though, when you scrub plants, so say you know the plant well, and you have a very predictable outcome in terms of how the human perceives it, not from a plant perspective, but from a human perspective. And you scrub it, and you take it back to its root. The stress of its lifespan epigenetically is adjusting it. And when you scrub it, sometimes you scrub the very things that people like to love. Like, that's some weird shit, but it happens too.
Not only the people like, but the microbes like.
Yeah, and so it's tricky because, like, I thought when I started scrubbing all my plants, I was gonna get this identical thing, but like refreshed. And what I did is I got incredible vigor boost, and I got it, pathogenically pure. But I also, on some of them, it changed profile because I realized the accumulated stress changed the plant and how it responds. And that's what I liked, was that stressed out plant response.
In that generation.
Yeah.
But second generation or third generation, that stress response.
This is the 90s. So who knows? I just knew that when we cleaned it, it changed. And not the bad way, but it changed. And if you're someone who focuses on the human element of how people consume and what they like and how they like it, you know.
Yeah.
So it's how when we clean genes, when we take away the stress, the metabolites that are produced as a result of that baggage also go.
So science is about doing things and repeating it and repeating it. So what Kevin's describing is a form of morphology. The plants evolve into something a little bit different under our own eyes. Are you referring to nodal cleaning and washing?
I don't care how you describe it. Yeah, no, because
Or meristem?
True. Nodal propagation is just propagation.
And washing.
The meristem propagation, where we're accelerating the genetic tissues and we're removing that tissue in pure form, it is a different plant sometimes. Not always, but some, and I'm talking subtly. If you're someone who lives with a plant for decades, you have an expectation.
And he has.
And a lot of people have. But it's just like...
He has.
It's weird, but sometimes when you clean a plant, the unclean plant has a higher marketability to the consumer, just less growability to you as the cultivator.
Could I give you an example? So I have a cut of Capulator's Mac 1. I don't know anybody who's ever grown it, but it's a particularly very difficult plant to get through the vegetative process. Let's just leave it at that. If I give you 2 tips, get it dry, get the roots searching out for water, the wider the base, the wider the plant. That's beneficial for yield. The second thing is don't top it, Charlie Brown it. Get it up there, let it grow what it does naturally. Strong like bull. But I took the Mac 1 3 years ago and began to process it through tissue culture. Every 3 months, all of my plants go through tissue culture. They are tested for hop latent viroid going into tissue culture, and they're tested for hop latent viroid coming out of tissue culture. So that at least I got a base point to say that, much like Kevin, my nursery is fairly clean. Every once in a while, at about a rate of 4%, some plants show up dirty. If you can explain that to me, I'd welcome you to come up and do it, because it could save me a shit ton of hassle. But as I work through the process and work towards a vaccine, I would hope, okay, the Mac 1 used to grow these really thick, dark green leaves. If you looked at that plant, you could pick it out of 5,000 phenotypes of all other plants. It looked different. When it went into flower, it flowered differently. Now that it has gone through tissue culture it is one of my oldest cultivars, so it's gone through tissue culture for the past 3 years at the rate of once every 3 months. So that would be 4 times 3, somebody give me an answer, 12. 12 times through tissue culture. Now the leaves are elongating. They're growing more sativa-like almost, okay? And the plant is more vigorous by 50 to 75% in veg. And once it reaches flower, what I thought was impressive has now become excessive. I'm talking, I need to cut my veg time down, I need to reduce the amount of light pressure that I'm applying, and I need to, of course, adapt to the changes within the environment.
And you touched on something really important there, because one of the things up here is public health and safety, and the change of a plant that came out of scrub, right? And you saw the morphological change, whatever it might happen. I have a question for you guys. Like, will that change the actual genotype of the plant, the chemical profile of the plant? Because we're talking about medicine here, right? How do we have more consumers coming into the marketplace, and how do they trust the genetics, especially if we're coming, into a new market where more things are getting scrubbed, like maybe an OG or a sour or whatever it might be, maybe another landrace that has some crazy cannabinoid profiles that are medicinally helping people's lives. So I guess, is that a kind of feeding off of that question? How has it changed, and will it change as we go through more scrubbing processes, if like that?
In my experience, I've cleaned a lot of different genetics. We're up to over 500 now, different unique varieties. Old stuff, Skunk One, Sour Diesel, Super Silver Haze, things like that. I noticed that they're better and louder and more vigorous. I haven't seen any changes, right? What I think some people might call changes are our cultivation practices are much more advanced now compared to back in the day, and that probably contributes a lot to it as well. And also, your palate changes every 3 months, right? So what was 10 years ago, with all the pathogen pressure now and all the different grow environment changing, I wonder if there's some bias in there regarding that, because I've cleaned stuff for people that remember something a specific way, and they get it back like it's the best it's ever been. I haven't heard anybody tell me that they've seen differences, and I wouldn't discount that it does happen, right? I just haven't observed it in the amount of stuff we've done, right? And also, when it comes to the science side of it, nobody has enough data to compare pre-tissue culture sequencing, tissue culture sequencing, post-flower sequencing, and comparing and looking, are there any differences, any slight mutations, epigenetic differences, right? And the cost to do that alone, to be able to say what is or is not, right, would be insane. Nobody's going to spend the money to do that and do a good job at proving or disproving or having that, in my opinion.
Good.
So we are doing science, but we are always thinking as a consumers. Imagine that you are a wine consumer and you love Cabernet Sauvignon. But the problem is that Cabernet Sauvignon in Napa is Cabernet Sauvignon, but the Cabernet Sauvignon in La Rioja, Spain, is Pinot Noir. And Cabernet Sauvignon in Napa, in Bordeaux, France, it's Merlot. That creates confusion to the consumers. to the audience, right? What about if you have an authentication method, a label that says Cabernet Sauvignon in Napa is Cabernet Sauvignon in La Rioja, Spain, and Cabernet Sauvignon in La Rioja, Spain is Cabernet Sauvignon in Bordeaux, France, or South Africa, or Australia, or Merlot, whatever. Why not include that? That is going to be safety for the consumer. That is going to be a proof of identification or safety, right? So this is what we are pretending to do to bring this extra layer of security to the consumer.
And education.
And education.
I think
yes.
a lot of the newer consumers know what they don't want to try again, right? I think that's the easiest way to help us understand when we start to learn a specific profile, fruit, floral, fuel, or earth, right, Kev? Like, how do we really lean into certain things? And those profiles, I know what I'm not taking or what I'm going to try before I go to sleep. A fruity or a floral, for me, that's going to keep me up. Fuel, earthy, heavy, right before bed, perfect. So just understanding what works for me, like I'm not gonna dive too deep during the daytime on some of those profiles that are gonna put me to sleep, but I'll still enjoy them. I just know how to react, how my body personally reacts to those. So a lot of these different things that have come over the years, training the nose, right? The nose knows, and then especially coming back to a trust factor. Public health and safety, right? How are we gonna get to a point in the industry where we have a genetic fingerprint that's on the label of whatever you're buying? Is this Blue Dream? Is this sour diesel? Because it's medicine. How can we get there? And these are the conversations I want to have because this is how we can start to elevate. Let's set the new standard for that. So I'm really excited about these conversations here. And Todd, you have a lab, DigiLabs. So you've seen the chemical profiles over the years. You know, what are some of the things that you're starting to see as you get more batch tests? Are you seeing any trends, any similarities to the newer markets coming online?
You know, we do see a lot of different varieties in the back. Lots of people are testing genetics all the time, always hunting, looking for the best, the brightest, the stronger, the fastest. We did a study where we took 2,700 samples from 206 growers and from 97 growers with 206 strain names. And we did the chemofile testing with cannabinoids and terpenes, right? And we looked at the data. And the data said that cannabinoids were pretty much the same across the board. And then we looked at the data on the terpenes. And the terpenes gave us 3 separate bunches, which told us that there's really only 3 main strains in Nevada with offshoots of everything else. Then we got the genetics tested on those, and it confirmed that the terpene identification really determines what the genes look like, right? I mean, you guys look for the terpene profile that you want in order when you're looking for the right genome, don't you?
Kinda. Under different production stresses, you'll produce a different secondary metabolite package. So I have to kind of compare apples to apples. So if I do it all indoor, or I do it all outdoor, I do it all mixed light, then I can say that I should have a consistent impact, standardizing, lets me measure. But like, to me, metabolites move, so you can see it in clusters and pictures, but it's a difficult thing to prove as a print unless you standardize and stabilize how you do it. Yeah.
I mean, the print is the ultimate, yes, it is what it is, right? And over time, they should be on the COA. Right? It's an honor for the cultivator to say, I have this certificate that says I have the real deal.
So let me understand. So you started the experiment with 2,000 samples. From those 2,000 samples, you got 200 names, right? How many genetic groups did you get? Like branches?
We got
4. 500 samples with different names, they were exactly the same.
That's exactly right. That's exactly right.
500 samples, genetically they are one.
That's right. I've been in a cultivation picking up a product to take back to the lab, right? And you know how proud cultivators are. And he brought it out and said, well, this is my Girl Scout cookies, but it really didn't come out as great, so let's just call it cookie something. That's how strain names are, as you guys all know.
So you have a tissue culture lab and you believe that you have 500 different strains, maybe you only have one. Maybe. Right now you have the tools to find out if it's right or not.
How different are the genes, right? Where is the genetic differences, and what makes a gelato a gelato, and what makes the OG.
I think we had a couple questions back here. Yes, right over here, sir. Name and where you're from.
Victor out of Hawaii, Big Island grown. So I do a lot of genetic breeding. I deal with a lot of Hawaiian heirloom cultivars and trying to kind of perpetuate and keep some of those things around because we are in Hawaii and try to keep true to that experience. I have found because I do have to meet quotas, there's things that I have to do. Do I perpetuate the preservation of the genetic true to the genetic, or do I water down to achieve goals while selectively hunting to keep true to the Hawaiian genetic, if that makes sense?
Yeah, both. You gotta keep a pool pure, so you can go back into it because you don't know until you're going down the track that you take the genes with you on this voyage. And if you haven't, then you have to go back into that unfiltered parental stock and open that up again. And the stuff that you're working with is rare because the populations got thinned. And so you want to hold as much genetic diversity as possible in that gene pool. And when you're able to have that as your mind, and then you take it and hybridize it to fit the realities of the market. But you can't throw away the parental population. That's it. Yeah, that's your safety base. So I do preservation work, which means I hold stock, and then I hybridize the hell out of it so that I can chase it. And if I am losing something, I can go back into that parental stock and say, maybe I need to open up a bigger population to find the plants that actually have the traits that are going to pass on in the progeny.
It's a great question, bro. Thank you. What else? Right here.
So you guys touched on a bunch of different points that kind of relate to each other in terms of like genetic fingerprinting, chemovars, terpene profiles, and public safety, labeling conventions and stuff like that. And all that kind of speaks to a certain finite number of like how many of these different chemovars, how many different product labels can a consumer read before there's like a confusion and they're not actually getting the medicine that they want or the product that they want or the reliability in the cultivar or whatever. Like because we're seeing so much overlap, and you see it in all these different components here. So it's like where should we all be focused? 10 chemovars that address all these conditions, 100 chemovars, 10,000 chemovars, and then how do we kind of bring that to a consensus as an industry so that we can address all that cannabis can be without so many different lanes that we can't kind of find a path forward?
I think that is a very great question. I don't think there is an answer yet. I have heard people that they would like cannabis be differentiated as ibuprofen. So you need ibuprofen and you don't care the brand, right? You go to Australia, you need ibuprofen. So you need THC with this specific percentage. And I think it's totally valid. However, you use the wine as an example. You love Cabernet Sauvignon. And this is Cabernet Sauvignon, not Merlot. I'm sorry, it's different. Right? So this is cultivar specific or related to your taste, right? So that is a big debate that hopefully the whole industry have an answer and learn.
And that's a really interesting point, but out of the entire spectrum of all wine, how many cultivars are used? And they're clonally produced to make all wine that we consume.
They are clones. They are clones from the same cultivars.
But like, he and I have an agreement on on diversity, where he saw 4 clusters, and I know that 4 clusters exist. So we use that as a compass. That compass allows people to make a big picture decision. Within each one of those big picture decisions, there's unlimited complexity, because at the end of the day, we're not all going to stimulate the same. But you can say, listen, it breaks up into 4 major divisions. That simplifies it. Instead of looking at 400, I can say 400 are really 100 of 4 different types. And now I can focus on one segment, one group. And the ability to simplify the process is imperative not just for the customer, but for the seller. So we're not like hustling pot. So if somebody's selling it to you, it's trying to give them the ability to be able to make good decisions quickly and easily, because people that are getting paid minimum wage for a dispensary job, it's hard to get them at this. They need to be super geniuses in differentiation. So you have to make it as simple as possible, but accurate. And I think that you can break it up into 4, and then from there it becomes the mission of the seller, and start to figure out where is their satiety. And then once they know that, I recommend, if you call possible, get the clone and then bank it. So this way, once you have something that works for you unbelievably well, it should work for a long time. And so medical cannabis is different than recreational cannabis in that regard. That once you find what works and stimulates, it's your responsibility to be in control of that process.
So something going along with the genetics, what you're talking about, I sell a lot of genetics all around the world, right? And I noticed a lot of the cultivators that I talk to, the first thing that we talk about is what's their market demand, right? At the dispensary, where they're selling, what do those folks ask for? And different regions have different demands. But for me, it's come down to kind of 3 different types, 3 different markets. Right now, California, hype, West Coast stuff, purple candy gas. That's what is like, I call like the recent Budweiser movement, right? That's what everybody wants, just at scale. You go to like the Midwest, a lot of the old heads there and people going to the dispensaries, they want Blue Dream, Sour Diesel, OG, a lot of that older kind of but well-known named stuff, right? And then go even further like East Coast, people are starting to go, oh, I want some Piff, I want some Haze, I want, and they're trying to be a little more different, unique, and varietal in that. So that's what I'm noticing those trends are, and the markets are dictating what genetics are demanded. And we try to work with the cultivators on providing stuff that fits the market demand, but is also good for the cultivator to grow in a financial model. And it's interesting to watch it all play out from this aspect.
Mr. Mojave Richmond, great question.
Justin, so your clients from South Africa, do they ask you what is the customer demand?
The South African demand?
For example, Australia.
Oh yeah.
You have no idea what is the market demand in South Africa. So
yeah.
Why they ask you that question?
Yeah, so I like exporting to South Africa. I phytosanitary exported genetics to South Africa. They grow there. It's a very interesting market. The wages are very low, so growing there is very cheap to do, and the product can sell at a good price. There's big margins compared to like growing in California, right? But talking with them, I had to like work through genetics with them. Like, oh, well, we just want like this name and this name. They didn't know what they actually wanted out of the genetics. They just wanted what people were asking for, So I had to work through, okay, well, when they say this, what do they mean, right? And what does that demand actually mean? And ended up selling them mostly old school stuff, like the OGs and the Sour Diesels and even Skunk One and some of that stuff, While we threw in a few of the new hype stuff to see how it does. And we're going to see. We're kind of like filling that market out. But what's interesting is there's a big market demand in England, UK, right? And those guys, there's a deficiency right now in the products out there. So they're starting to go around and they're starting to fly to South Africa to actually bring back. And they're manipulating the market with that market demand specifically. So the cultivators are starting to ask me, we're like, oh, I want Gary Payton or Fish Scale or all these, new different hype stuff. And they don't know what they're asking for, but their buyers are asking for it. So
it's getting infiltrated.
Yeah.
It's a great question, Mojave. Thank you. What other questions? What do you say, guys? Should we wrap it up?
Yeah.
Any other final thoughts, questions, curiosities?
Realize that.
That's right. Amen to that. It's about good people, spending your time with good people with good intentions. That's what the Connect community is all about. Inspiring, educating, leveling each other up. Let's grow together, guys.